Our session was a mix between preclinical work and clinical work. That means working on models of cancer, not in patients, and actually data coming out from cancer patients treated with different immunotherapeutic agents. We moved from the early stage clinical and preclinical data on molecules or antibodies that target molecules such as CTLA-4 and PD-1, and their movement from understanding how they work in vivo to actually clinical trials demonstrating efficacy against melanoma. So those were some of the findings that were discussed during my session. There was a lot of work also and discussion along the lines of a new target called CD137. So this is a molecule expressed on the innate cells of the immune system, the natural killer cells. And what Professor Holbrook-Court demonstrated in preclinical models and also in the clinic is that antibodies that target this pathway can synergize with, for example, antibodies such as rituximab and produce systemic tumor destruction and actually activation of the natural killer cells to promote further destruction of tumors. So that was a very, very interesting session. And then Rodrigo Selenay from the Manchester Cancer Institute had some very provoking data in preclinical models of cancer, demonstrated actually that there is a pathway named the COX pathway that prevents immune reactivity against tumors. What's very interesting is that the COX pathway is targetable by molecules such as aspirin. So he demonstrated using mouse models of cancer that if he combined aspirin treatment with antibodies against the PD-1 pathway, he gets maximal anti-tumor activity. So that was very interesting. And then the last speaker of the session was Emanuela Romano, and Emanuela had very interesting data showing that in vivo, in humans actually, the anti-CTLA-4 antibodies that is being administered to patients actually has a dual activity. It has an immunomodulatory activity, which is what has been described originally. It releases the brakes on immune cells and get them very, very active against the cancer. And a secondary activity, which is actually the ability of killing a subset of immune cells called regulatory cells. So actually in one single antibody, you get two activities, the immunomodulatory activity and then the Treg depleting activity. And that was pretty much what we had on the session. So it was a mix between different targets, different types of tumors, and actually preclinical data and clinical data. So I thought it was a very exciting session. What was solid throughout this session, it was a constant desire from these investigators to understand how are these drugs really working. And by figuring that out, they can actually now predict how other drugs might work and actually try to improve the functionality of these drugs by better combinations or further engineering of the drugs that they're using. So it's all about mechanisms. But I'm very biased because I believe that mechanisms are going to tell us how to improve our pipeline of therapeutics. It tells them a little bit about the fact that PD-1 and CTLA-4 are here to stay. That's one thing. There are issues with those drugs because they induce toxicities and they need to be managed. But actually, they're not the only ones on the horizon. The horizon is packed with a large number of other drugs that will also help mobilize immunity, and not only immune modulatory drugs like new anti-CD137, et cetera, but actually old school drugs, perhaps even like aspirin, right, that could help change the tumor microenvironment and make it a little bit more permissive for immune modulation. We're having drugs that are really impressive in their activity, but they're also very expensive. We didn't discuss too much of this in our session, although Holbrook did touch on it. These drugs, per course of treatment, they are higher than 100,000 US dollars. So there will be complication there. We're going forward with more combinations to increase responses, but those combinations are going to have increased costs. So I think that how are we going to deal with that? I don't know, actually. I think that's going to be an issue that pharma will need to resolve, that obviously the NHS will need to get involved at some point. And as a scientist, all we can say is that these drugs work. But how are we going to implement the policies to deliver them to patients that really need them at a costly basis without breaking the bank? That's going to be a subject of a huge, larger discussion. So I think for patients, what I can say is that there is a huge effort on the scientific and clinical side to really understand how these drugs work, and actually when they don't work, why is it that they're not working? There is a huge degree of hope in the future, I think, because again, PD-1 and CTLA-4 are not the only drugs. This is just the beginning, and actually we're seeing right now the results of 5 and 10 years of research, and the research that is being conducted right now which we're not talking about it yet because it's not ready, it's not mature, that's going to generate the next 5 years of exciting data. So for patients, all I can say is keep an eye for strong scientific rationale when it comes to combinations, and there is a lot of strong scientific rationale behind many of the combination therapeutics that are being implemented now in clinical trials.